Yeah, when you send for sequencing of any DNA using Sanger sequencing method, so you will get two files. One is this SUQ file, which you can see here, and the other one is AB1 file. So the same thing, same for a same sequence, you will get two. So here, see, the code is the same, A, and this is also A. In fact, the same sequence, but here you can open in a notepad. It will have only the sequence information, nothing else, just a sequence. So that's why it's a very small file. So it will not have any other information, just a nucleotide sequence you can see. Okay. But here the same one, it will have more information. That's why this 232 KB. In fact, here is just 1 KB, which is just nucleotide alphabets only. You'll have the more information you can see in the AB1 file. Both files you will get once you send for sequencing using Sanger sequencing method. This Sanger sequencing method, as I explained to you, the technology you understood. So it is using the dideoxy, and when you are using the automated uh, genome analyzer, so it will have uh, automated robotic uh, tips which can collect the sample into the gel, and gel in the sense capillary gels, and it will migrate according to the sizes and the colors, and the fluorescence dyes will be detected by the uh, detector, and that will be decoded into the nucleotide respective nucleotide sequence automatically. Okay, so that's so normally, if not automated mission, so every sequence you need to run a big gel. So A separately T, that means DDATP, DDTTP, DDCTP, DDGTP. So four wells you need to separately you run, and a very big gel you need to cast and load the gel, and then you need to remove the gel and expose to the X-ray film, and the next day you will get the bands. Then you have to analyze the bands manually, and you have to find the sequence. But slowly it took a few years to get automated. Still we are using that automated machine for the Sanger sequencing for the small scale. If you don't want whole genome, just for cloning purposes, 1 2 or 10 20 sequences, you still use the Sangers only. But the what we are going to learn today is next generation sequencing, where it will start with the pyro sequencing as drastic improving the technology. So in the Sanger sequencing, as I understood, at a time, normal with advanced technology, you can use 96 well plate, and thus you can sequence the 96 DNA sequences at the same time. But using the new technologies, we are going to see the next few, I mean, 2 3 classes, this Roche pyrosequencing and Illumina Solexa sequencing. So these two technology we will more understand, and this one we will not go into deeper. Okay. So this is the at present Illumina is the most using sequencing technologies for genome sequencing as well as transcriptome sequencing. And Pyro, still few labs use it. This is a transit from the Sangers to uh, the really now at present Illumina. This is a Roche mission which uses the Pyro sequencing, entirely different from the Illumina sequencing. So we will understand what is Pyro today. And we do have a heliscope, helicos technology, and smart by Pacific Biosciences. At present, this is also getting popular at present. And This one to compare what you learned, this capillary based applied biosystems um, Sanger sequencing, this one. So you will get approximately around 700 nucleotide sequence of a single DNA sequence at a time, but you will not get too many sequences. So data output data in few kbs, that means 96 samples you can sequence. So you will get some few kb of data. I mean 96 times around 700. So that much data only you can get few kbs for one run, the output of the data. But now what we are going to see that 454 are row sequencing. So the sequence length is only maximum 300 to 400, around 300. Okay. And but it's not only 96, you can sequence few thousands and or few tens of thousands at a time. So you will get millions of base pair sequences you will get at for one run. Okay, for one sequencing run, you will get some millions of sequences. And Illumina is also from Applied Biosciences, and this one previously it was when it started only 50 base pairs of any sequence only it can go, but now this also can sequence until around 400 to 500 nucleotides for each one you can sequence. But this output of data one run will give giga base pair, so GB you will get, so which is very huge of amount of data you will get for one run. Okay, so for Pyro sequencing, you need to first prepare a library. So we'll discuss in a while. And this was first published, this technology first published in 2005 and modified in 2006 in Nature. This is one of the milestone in the sequencing technologies. And this picture describes the whole flow of pyro sequencing. This picture alone is enough, but we need to understand each one what is going on every step. And I'll try to explain to you every step and try to listen. I'll use my whiteboard for explaining. So let me I'll stop sharing so that you can have bigger view. See or not. Okay, what is the first step? We need to do the library preparation. See here. Okay, you can see it. So how to do the library preparation? First, you need to take the genome or DNA. DNA, right? So this one you need to break into small pieces. So around 300 base pairs. Each one should be around 300 base pairs. This breaking, just a minute. So this is around 300 base pairs. So you will have millions of these kind of sequences when you when you break the whole genome. Okay. So after that, what you are going to do, you are going to add some adapters of the 5' and the 3'. So you are adding some adapter. So this adapters are nothing but a DNA sequence. So this is you are adding at 5' and 3'. So in genetic engineering, you have understand a little bit about it, how to do the ligation of the DNA and how to lyse, isn't it? So some of the enzymes can break, some of the enzymes can attach. So using those different kinds of enzymes, so we are doing this, adding some, with breaking the sequence around 300 base pairs and adding the adapters at the 5' and the 3'. So the next, what we are doing, so you understand so far, right? So whole DNA or genome sequence, we are breaking into small pieces, which is around 300 base pairs. And then you can select it, especially 300 by running on the gel. So run on the agarose gel. So you can use the ladder. So 100, 200, 300, 400, 500. Like that, you will have the ladder of known DNA sequences, and you run your sample when it is coming around the 300 to 400 somewhere here. And if there is any undigested DNA or small DNA, you need to exclude. So cut this gel and purify this gel, and that will have around 300 base pairs of all the sequences, and that sequence you take and add the adapters at the both ends. So you will have millions of different DNA, but having the adapter is the same adapter at the 5' and 3'. Okay, so this is the first step in the Pyro sequencing. So what is the next one? Amplification. So here we are okay. The second one, what we are having this the emulsified PCR. So we are going to amplify those fragments, DNA fragments. Those DNA fragments which are having around 300 and having the adapters. So we are going to amplify them. How we do amplify? Not normal PCR. We are going to use a PCR tube which is having the emulsified solution. So you know the emulsion, right? So which is having the oil droplets. So it is having the two phases, aqueous phase and organic phase. So these oil droplets, it will be having in the PCR solution, and this will have some millions of droplets, and you add these all the DNA to this solution in a ratio. It is standardized such a way that each one will go and enter into the one emulsion, one oil droplet. So this oil droplet, each oil droplet contains some beads, so which are having some immobilized oligos or primers. Immobilized in the sense they are covalently bound, so you cannot detach them. So it is covalently bound and it is immobilized. So again I repeat, there is emulsion solution. This emulsion will have oil droplets. So each oil droplets will having some beads, and each bead are having few tens of thousands of immobilized oligos are the primers. Okay, and this emulsion, inside this emulsion, that is inside the droplet, oil droplet, contains all the reagents of PCR. So you understood already what is PCR, right? So all the ingredients of PCR are present in this oil droplet, individual oil droplet, and each droplet is having a lot of beads which are having immobilized primers. So another thing here need to be understand that this oligos are complementary. These oligos are complementary to this adapter. These oligos are complementary to this adapter. What happens when you take this sequences, add it to this emulsion, and it will go enter into the oil droplets, and it will enter into the, after entering the oil droplet, it will bind to this complementary sequence, isn't it? It will go and bind to the its complementary sequence. So the ratio of the DNAs should be such a way it can go enter into the oil droplets and bind to only one bead at a time. So only one bead. So it will immobilizely present one at a time. But few can present two, few may not have anything. Okay. So after entering the DNA into the emulsion and into the droplets, in the droplets, it will bind each sequence, one sequence bind to the one bead, one sequence bind to the one bead. There are some beads not bound to anyone. There are some beads bind to the two sequences. But most of them, this is a max, like this. This kind of things will find maximum possibilities. So the ratio calculated such a way. So I hope until this is clear. So the next one is you do the PCR. So this one you keep into the PCR mission. It will be slightly different from the normal PCR. So this one will amplify. So how to amplify? So DNA polymerase already present. All the ingredients of PCR already I told you inside. What are those? It will be having DNA polymerase, then what else? DNA polymerase usually use the tag, but but this kind of things, so no need to be tag. So different DNA polymerase. So DNTPs, what are those? DATP, DCTP, DGTP, and DTTP. So all will be there. So what happens when the DNA polymerase is there, DNA template also here, DNTPs are here, oligo or primer is bound to the one end. So it just amplify. So it just amplify the other strand. It just amplify the other strand. Okay. So then similarly, after this one, so you denature and again anneal. So this will denature and anneal. So it will go and amplify every place. So this one is amplified. This is complementary to the this sequence. Okay. So what you will get now, you will have the beads which are having your sequence, of course, complementary sequence. I mean, almost the same, right? So you are having a multiple copies of the same sequence. At the end, what you will get? Multiple copies of the same sequence in the each droplet. What you are having. Okay. So by this, the second step finished. That means what is the second step you are doing? You are introducing the small fragments of the DNA which is attached to the um, adapters and to the emulsion PCR solution. In emulsion PCR solution, it will be having the oil droplets. Each oil droplet contains all the ingredients needed for the PCR, and also it is having some beads which are immobilizedly present, multiple copies of immobilizedly present oligos which are complementary to the adapter which you introduced in the first step. And once you introduce and amplify multiple cycles of PCR, you will get thousands of the same sequence present on the bead. So they are immobilizedly now present because as oligos are immobilizedly present, and the extension of it also, it will be strongly stick to the beads. So you cannot detach them. So thus the second step completes. Then the third step of the pyro sequencing. In the third step, so you will be having a micro plate, so chip kind of thing, which is having a plate. Few layers will be there on it. Few layers. And on the top, it will be having some holes. Just to show you a big holes I'm keeping, but it is having millions of small holes which you cannot see manually with your eye. So thus everything here is automated. So you are not going to do manually anything. So it will be automated. So what you are going to do, so these beads, what you are having, all these you are introduced to these plates. So the hole size, the pore size of the hole is such a way it can take only one bead. It can take only one bead. So you can imagine something like this. It can able to take one bead. Like this. Okay, so millions of these holes will be there on the chip, and a whole PCR after that emulsion you are going to introduce to the plate, and such a way, I mean, it will take only one bead into the one pore. So some of the pores can be empty, but maximum one only can go into the one pore or hole. Okay. So this is the intermediate between the second and the third step. So after introducing to this, we are going to add few more small beads. We are going to add few more small beads to it. So I'll tell you what are those beads later. You're going to have a small beads. Then apart from that, in the third step, what we are going to do, this is how it looks like. Maybe I'll show you the slide so that you can. Yeah, you can see here. So the first step, you already cut the DNA into small pieces. Then this one you introduced into the emulsion, where having oil droplets. Then oil droplets is having, each oil droplet is having the beads, and beads are having immobilized primers. And once it attach, it makes multiple copies of the same. And after that, so you introduce to this chip or plate. So each pore can take one bead only, here you can see. And after that, you will be having added a lot of small beads. Okay. So this is how it looks like under the electron microscope. Okay. The next step is, let me show you the slide first, the next step, then I will explain on the board. So this is how it looks like, the chips, which you cannot see any pores, but it is having some millions of it, micro titer plate. Okay. These reaction will occur in the next step. What are those? You are adding all the beads which is having the nucleotide, and you will also add some primers which is complementary to the adapter, and you will add the polymerase. We'll add ATP sulfurase, you will add luciferase and luciferin, APS. So why am I giving you all this at a time? Because those beads should contain all those which are needed for the next reactions. These are the three reactions will occur. So those small beads which I have explained here, these beads will contain DNA polymerase, some of the beads. Some of the beads contain ATP sulfurylase. Some of the beads carry DNA polymerase. Some of the beads carries ATP sulfurylase. Some beads will have luciferase. Apart from that, you will add the substrate luciferin and APS. Okay. So the next thing, what happens, the reaction, the main reaction going to be happen once you introduce the beads to the plate. So those picotiter plate will be placed into the different mission, where it will do the pyro sequencing reaction and sequence deciphering. Okay. So what it will do? So okay. So the first reaction is this nucleotide, the nucleotide sequences, all which are, everything is similar, which we have understood, right? All are same sequence. You add a DNA sequence which act as a primer and which will attach to the other end of the DNA. So how you can add, how you know the sequence, because you will have another adapter here. So you just need to be added the complementary sequence to the other end. So it will go and bind to the DNA, which is complementary other ends. Okay. So you added the DNA. So after adding the DNA, so you add DNA polymerase, which are carrying these beads. DNA polymerase. So what happens here? So you will have a DNA. This is the bead, which is immobilizedly present. DNA sequence you will have, and the other end you will have the primer, which is complementary attached to that. So after that, what you are going to do, you are going to add a nucleotide at a time, one nucleotide at a time. So insert this chip into the mission, right? So you pump DNTPs at a time. So let's say you added. So this is the sequence. Just to appear, I just written with bigger letters. This is around 300 nucleotides, right? Okay. So now what you do, what you are going to do, so do this, the whole chip, whole plate, we are injecting D, T, T, P, D, T, T, P. So what happens when you add DTTP and DNA polymerase is also there, DNA polymerase also there, and DTTP is here. What happens? This A, which is complementary, that is T, it will be added to the primer. So this elongating primer, T will be added. I hope it's clear for you. So T is added to the primer because A is the complementary to A is T. So just like your PCR, so it is from the primer, T is added because you added to the whole chip DTTPs only. Only one nucleotides only you are adding, not all four. Okay, so try to understand. So though elongation is similar to the PCR, that is DNA polymerase is adding, but it will not add all the nucleotides because the C, G, which are needed to the elongate other things is not there. Only you are added only T. You are not added anything else now. So only DTTPs you are added because of that T is added over there. So if you understand the reaction, how the polymerization occurs, so it will be added the 5' the triphosphate to the 3' hydroxy to form a phosphodiester bond. During the phosphodiester bond, you remember the pyrophosphate will be released. Do you remember? So this phosphate is going to have a bond with here, and this pyrophosphate will be released. This is the very important information in this pyro sequencing methodology. Once you add a T, PP, pyrophosphate, is released. So what happens once it releases the pyrophosphate? So come to the reaction in the slide. The pyrophosphate is released. once you add the nucleotide, pyrophosphate is released in presence of polymerase. This pyrophosphate reacts with APase, which is added to the beads, to the small beads, which acts as a substrate in presence of ATP sulfurylase, which also you added to the chip, already present in the beads. Okay, so ATP sulfurylase also present to the beads, and APS also present in the beads. The soon after it releases the pyrophosphate. Pyrophosphate combines with APS, and catalyzed by ATP sulfurylase, it produces ATP and sulfate. Okay, so I hope you are following. Hello, any difficulties so far now? Students, are you following?
Yes, sir.
Okay. So pyrophosphate, which released in the first step, that means when you added the one nucleotide at a time. So I gave an example of as a DTTPs, and then it will react with the APS, and there's, I think, something adenosine. APS stands for something. Adenosine something. I check and let you know. It should not be ammonium persulfate. It's not ammonium persulfate, because see what happening here. You understand the reaction. Automatically you can guess what it will be, because ATP is producing. ATP is adenosine triphosphate, right? So triphosphate, the two phosphates are coming from here, pyrophosphate, and it should be something adenosine. I'll check and let you know. And then ATP sulfurylase. So yeah, then it will produce ATP and sulfate. So here is the important thing. What you got here, ATP. So ATP, one ATP is produced whenever one nucleotide added. See, indirectly, what is happening? One nucleotide added, one T is added to the, for example, T is added to the primer, and one ATP produced. How? Because it produced one PPI become one ATP. Okay, this ATP again acts on luciferin, on another substrate which is present on the beads. And of course, luciferase, another enzyme also present in the beads, and oxygen is present in the environment, surrounding environment. And so the luciferin will be converted into oxyluciferin. This is the product, and luciferase is an enzyme. ATP is utilized for this reaction. When there is no ATP, oxyluciferin will be not formed. When there is a ATP, one oxyluciferin will be formed from one luciferin. Okay, and apart from that, oxyluciferin is produced. It's not actually our interest. What is our interest? Apart from the oxyluciferin, the ATP is actually broke down into AMP and PPI, and also oxygen is released as carbon dioxide, and this reaction release the energy in the form of light. This light is our interest. So what happening now? Whenever the nucleotide added to the primer, so light is producing. So that is what you need to understand with this reaction. So let us come to the here the well beads what we are discussing. So when here 1T is added, whenever 1T is added, first reaction pyrophosphate will be released. Whenever the pyrophosphate is released, this pyrophosphate will combine and gives ATP. So the second reaction, okay? So APS plus PPI in presence of ATP sulfurylase, then it will give ATP. This ATP utilized in the third reaction where luciferin will be converted into oxyluciferin, ATP utilized, luciferase, and plus light. So light is important here. So what actually you got, whenever the whenever the nucleotide is added, light is released. So then this plate is actually visualizing by a light detector. Whenever any small well, any well is releasing the light, it will detect and record it. Okay. So now what we have done, we have added the TTP. We have added the TTP. Okay. The light is released. So light peak will be there. Okay. So the next one you add. So we put in a sequence. A TTP is added. The next one is, let's say you added DATP. So before adding DATP to the wells, you need to flush remaining TTP. So there will be washing step. So this washing step will inject some of the buffers which will remove the existing TTPs. So TTPs is erased from the well. All other enzymes are there. That's why we put the beads. Why we are not adding the enzyme separately? So in order to wash, when you wash, so only it should be removed the nucleotides, not others. So all other ingredients are present in the beads. So there will be no worry. Only it will be remove the TTPs, nucleotides. So now you inject again DATP. So when you add DATP, so will it be added to the elongating chain? Will it be
No.
added? No, right? So it will not be added because G required C as a complementary. So when you add A, there will be no addition of A here. So there is no addition, there is no PPA, there is no PPA, there is no ATP, there is no ATP, there is no light. So there is no light. When we add A, there is no light or just small background signal. That's it. So like otherwise, no, nothing. So no light. So then you add, so then again you wash here. So the machine, these are all automatic. So the robots will be doing, and we are not doing anything. So DCTP. Okay. In a sequence, we had like a DACG. Let's say there is a sequence. So the third one you added CTP after flushing off the remaining ATPs, DATPs. So C is added. So will it see added here? Yes or no?
Yes.
Yes. So C is added. When you see add, automatically all these reactions will leads to the light. And when you add C, there is a light. And then you flush this one, remove it, and you add DGTP. And when you add GTP, will it be added GTP here? G? Yes or no?
Yes.
Yes.
Okay, G will be added and the light will be produced and it will have P. So the next one again is the next cycle. So it's the sequencing not stop here. So you will remove this and add it again DTTP. So what happens? T will be added here or not? Will it be added? So it will not be added. So nothing. So T will be not added. Then A. When you add A, what happens? Will it be added or not?
Yes.
Yes. So thing is here, there is no control here. It will add another A also here. It's not only one A, two A's because two T's are there. So two PPIs will be produced and two ATPs, two times intensity of the light will be produced. So the light peak will be bigger, which is almost double to the normal. Okay. And when it comes to the T, of course, after that A, C, G. So CG also will not have any peak. Then the third cycle, when it comes to the T, it will form three T's. That means beak will be there when it is having T. So A, C, G, nothing. And then T will have a bigger peak. So this is how it will detect the light which are produced in this each well. So we are seeing the chip which is having millions of pores, and each pore light will be detected by the camera. So it will take information of each one, and each information will be stored separately. So for each dot, each well, there is one graph. Every well, so millions of sequences information will be generated parallelly. And after generating this graph, you can understand what is the sequence of it. Of course, software itself it will give you. So what is the sequence? By this, here it will be having TACG, TACG, TACG like that. But based on the peaks, you can understand T. That means it will be A. Okay, it is A. Then A is nothing. C, okay, G. Then G is 1, C. Then T is nothing. A is taking 2. So that means T, T. Then C nothing, G nothing, T is triple time of the graph. So this is a 1, this is a 2, this is a 3, this is a 4 peak. Okay. So then T triple times the peak. So A. See the same sequence which you got here, here we got it by the peak graph. We can get it. So for each dot, for each well or for each pore of the chip, will have one graph which is recorded based on the light, and millions of graphs it will generate by this camera, and then it will give the sequence of millions of sequences at a time. So that is the advantage of this pyro sequencing. I hope you understand. So this is the main sequencing reactions. So oligonucleotides are there. At a time, one nucleotide will be added. For every nucleotide added, a pyrophosphate will be released. And then this pyrophosphate reacts with APS to release ATP. This ATP utilized further luciferin conversion to oxyluciferin, where the light will be released. So this light will be detected, and Yeah. So the light intensity will be detected. So this is how the machine looks like. So you will be keeping the chip over here, and then it will be connected to the pipes. So where at a time the single nucleotide DNT piece will be injected, ATCG, which is placed here. And after each nucleotide, there will be a flushing, the washing step will be there. And this is how the light will be released, and the light will be detected. And this is the pyrosequencing results, how it looks like. So when it is a peak, this is the A, and this is a peak which is 4 times higher, that is 4Gs, and one time this is a T, and there is nothing here, our background signal, nothing here, nothing, and this is the GG. So the sequence is going to be A, GG, GG, T, GG, C, TTT, GG, GG, TT, GC, A, G, TT, G. This is the sequence you can decode from this graph. This is how it looks like, the each one in a different fluorescence color. So what are the advantages of this pyrosequencing? It is much more accurate. Whenever that particular nucleotide taken only, it will be release that the light. Parallel processing. So as no need to wait for another 300 nucleotides to sequence some time. So all can be sequenced at the same time, millions of sequences at the same time. So parallel processing. Runtime is 8 hours. So it's not many days or something, just 8 hours is enough, and easily automated, eliminates the need for the labeled primers and nucleotides. So you don't need to worry about the radioactivity, which is more harmful. So no radioactivity is needed. Mainly here, the light is there, so there is no side effects of it. It is much safer. No need of gel electrophoresis, unlike the Sanger sequencing. And read length is around the 300 to 400. Even though it is a millions of sequence you got and a short sequences, but short sequence is not that small, you can get around 300 to 400. And the disadvantages also there. Disadvantage is what? Small sequences. So you will not get like Sangers, which you can get 700 or up to 1000 also you will get, but here you'll get around 300 or maximum 400. And the another main problem occurs when you have redundant nucleotides, random repeat. So you saw AA, it is present double this, not really double the peak, but relatively high. When it is 4 times or 5 times is okay. Can you see, can you imagine when the one nucleotide is repeating like T, or T is repeating 10 times, T, 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 10 times. So the light intensity you cannot get 10 times higher than the normal. So it is a slightly variant, it will be lesser than that. So it will be it will be having some difficulty, because the light response to the nucleotide number is not linear. Okay, so that's why if it is more than 5 to 6 identical nucleotide, there is a little bit challenging. So apart from that, it is very good. And it has been using for de novo sequencing of the genome, variation detection, gene expression, metagenomics. And this is the first paper published using the pyrosequencing technology. Neanderthal genome DNA was sequenced in and published in 2006 and published in Science. So like after that, there are so many genomes have been sequenced using this pyrosequencing method. Any questions related to this class? Hello, have you understand the pyrosequencing?
Yes, sir.
Understand?